Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we'll talk about glycolysis pathway defects. In previous videos, we have established that the generation of ATP from glucose is perhaps the most important metabolic highway for human survival. This highway that we call Main Street is composed of glycolysis, PDH, Krebs cycle, and the electron transport chain coupled to oxidative phosphorylation. In the last video, we focused on glycolysis, important aspects of this metabolic pathway called glycolysis, also known as the Embden-Mayoff pathway. Going through the glycolysis pathway defects will help us apply glycolysis and understand its connections with human physiology. As mentioned previously, glycolysis is made up of 10 reactions, each reaction catalyzed by a different enzyme. Now, what happens if a certain enzyme is present in very, very small amounts, there is deficiency of that enzyme, or there's a complete absence of the enzyme, or the enzyme doesn't work? So for example, let's say someone doesn't have this enzyme, triose phosphate isomerase, which catalyzes reaction number 5. So if reaction number 5 does not happen, that means dihydroxyacetone phosphate will not be converted to glycerol 3-phosphate, and that will lead to the accumulation of DHAP. So basically, glycolysis will continue and stop here, and more and more and more DHAP will form. So the first consequence of not having a certain enzyme is the accumulation of a certain chemical. Now, a cell is already a very crowded space. Cells are very crowded. So whenever you hear the term accumulation, that's pathological. Okay, normally no chemical should accumulate in our cell. Accumulation means it's a pathological situation. It's not normal. So the accumulated chemical could interfere with the normal cell functions. Now, accumulation of chemicals will also increase the osmolarity of the cell. So when the intracellular osmolarity increases, then water from the outside will enter the cell and the cell will start swelling up. And the excess water in the cell is going to impair cellular functions. Another thing that could happen with the accumulation of chemical is that the accumulated chemical may be toxic by itself. So for example, dihydroxyacetone phosphate, if it's accumulated, we'll see later that in a disease called triose phosphate isomerase deficiency, DHAP is toxic by itself. It's a very toxic chemical because it has a ketone which is very reactive. Even if the chemical was not toxic, the chemical may be converted by other metabolic pathways into chemicals that may be toxic to the cell or to other cells. Now, if the enzyme, which is a protein, is misfolded, so it gives you a defective enzyme, so it doesn't fold properly. If it's misfolded, now misfolded proteins, including misfolded enzymes, can trigger a cellular response called an unfolded protein response. Now this unfolded protein response is a program that can lead to apoptosis. The cell can actually commit suicide by apoptosis. So in this way, enzyme defects can be quite dangerous to human survival. So with this background, let's look at some examples of the glycolytic pathway defects. aldolase A deficiency. So remember, the enzyme aldolase catalyzes reaction number 4, where fructose 1,6-bisphosphate, a 6-carbon molecule, is broken down into dihydroxyacetone phosphate, which is 3 carbons, and glycerol diphosphate, which is also a 3 carbons. So this is the breakdown of a larger molecule into 2 smaller molecules. There are various isoenzymes of aldolase. So aldolase A is mostly found in skeletal muscles and red blood cells. Okay, so this deficiency is aldolase A deficiency. Now, aldolase B is predominant in liver, kidneys, and small intestine, while aldolase C is predominant in the brain. Now, leukocytes, the predominant isoenzyme, aldolase isoenzyme in white blood cells, is also aldolase A. So in this disease, aldolase A deficiency, the aldolase A that is made in the cells is very sensitive to higher temperatures, for example, during a fever. And at higher temperatures, the enzyme is not very active. The catalytic function of the enzyme dramatically decreases. So in these individuals with this aldolase A deficiency, when they have an infection, their leukocytes function may be affected because the aldolase A in the leukocyte is not very active, so the leukocyte cannot generate much energy, and so its ability to fight the infection is reduced dramatically, and this leads to very frequent occurrences of fever. So during the episodes of the fever where the body temperature is higher than normal, the aldolase A becomes less active. And since aldolase A is found in skeletal muscles and in red blood cells, that lack of activity of aldolase A leads to insufficient energy generation in red blood cells and muscles. That leads to weaker red blood cells and muscle cells, and that causes breakdown of red blood cells, what we call hemolytic anemia, and breakdown of muscle cells, what we call rhabdomyolysis. So you can see that not generating sufficient energy, as in the example of aldolase A deficiency, can lead to impaired function of cells as well as to weaker cells, in this case weaker red blood cells and weaker muscle cells that tend to break down easily. The second example shown here is triose phosphate isomerase deficiency. Now, triose phosphate isomerase catalyzes reaction number 5. This disease is the most serious glycolytic disorder. Without this enzyme, or under small amounts of this enzyme, deficiencies of this enzyme, this reaction will not happen, and what we see is an accumulation of dihydroxyacetone phosphate in red blood cells, neuron, and in many other cell types. Now, if you look at dihydroxyacetone phosphate, it is a ketone and it has only 3 carbons, so it is too small to form a cyclic structure. Okay, so unlike glucose and fructose, which have 6 carbons, they can form cyclic structures and hide their ketones and aldehyde groups. Dihydroxyacetone phosphate cannot. So aldehydes and ketones are very reactive even in water. Therefore, the accumulated dihydroxyacetone phosphate will react with all kinds of cellular proteins as well as other small amines, because ketones and aldehydes react very, very well with amines under cellular conditions in water, 37 Celsius, and that leads to the formation of what we call advanced glycation end products, or AGEs. Something very similar we see in uncontrolled diabetes. These proteins that react with DHAP, what we call glycated proteins, they do not work as well, and the AGE chemicals are toxic, and all of these leads to problems with red blood cells, neurons, muscles, and other cells, and that results in the breakdown of red blood cells in the body, hemolysis, impaired nerve function, as well as impaired muscle function. So infants afflicted with this disease, they will have chronic anemia, hemolytic anemia, because the red blood cells are breaking. When red blood cells break down, we shall see in the future, the hemoglobin is converted to bilirubin. Bilirubin leads, high levels of bilirubin in the body leads to jaundice, which is the yellowing of eyes and skin. There is neuromuscular dysfunction, heart problems, and increased incidence of infections. These infants usually die of respiratory failure and or infections. In the disease phosphoglycerate kinase deficiency, this enzyme phosphoglycerate kinase, which catalyzes reaction number 7, is either absent or deficient or defective. The gene for this enzyme, PGK1, phosphoglycerate kinase 1, is located on the X chromosome. So the individuals who are affected by this disease are mostly males due to the X-linked recessive inheritance pattern of this gene. So without this enzyme phosphoglycerate kinase, once again glycolysis is affected and energy generation is affected. So there is reduced energy generation. And energy is very important for cells. So without enough energy, we can have two forms of the disease. One is the hemolytic form, where the red blood cells are seriously affected. In the second form, the myopathic form, the muscle cells are seriously affected. So in the hemolytic form, the red blood cells are very weak and they tend to break very easily. So that leads to chronic hemolytic anemia. So breaking of red blood cells means your red blood cell count will go down. So without enough red blood cells or anemia, the individuals will show symptoms of anemia that include pale skin, shortness of breath, as well as fatigue, because there is not enough oxygen that is being carried by the red blood cells in the body. The excessive breakdown of red blood cells will release hemoglobin, and the hemoglobin will be converted into bilirubin. And this chemical bilirubin is yellow, and it will lead to jaundice. In the myopathic form, the lack of energy generation in the muscles will lead to muscle weakness, pain, as well as cramping. And if this is untreated, it may lead to muscle breakdown. So the muscle cells will actually break down. If you remember, muscles are very rich in this protein called myoglobin. Myoglobin is found in muscles, and excessive loss of this protein from muscle cells will cause this protein to come out into the urine, what we call myoglobinuria, and excessive amounts of this myoglobin in the kidneys can actually lead to kidney injury, so kidney disease. Another glycolytic pathway defect is the phosphoglycerate mutase deficiency. This enzyme phosphoglycerate mutase catalyzes reaction number 8. So in this disease, the MM isoenzyme of phosphoglycerate mutase is affected, but not the BB isoenzyme that is found in red blood cells. So this disease will only affect muscles and not red blood cells. So without enough energy production in the muscle cells, we can have muscle weakness, pain, as well as cramping, and that can all lead to the breakdown of muscle cells. And as we mentioned before, muscle cells are very rich in myoglobin, and when these muscle cells are damaged or are broken, a lot of myoglobin will be released into the bloodstream. Now, myoglobin is a small protein, has a low molecular weight, so it can very easily go through the filtration system of the kidneys and come out into the urine. So the excretion of myoglobin into the urine is called myoglobinuria. Unfortunately, myoglobin can also precipitate in the kidneys. So this protein, excessive amounts under the right conditions, can also precipitate, and that can cause problems in the kidneys such as tubular obstruction, and that can lead to very serious kidney damage. Now, pyruvate kinase deficiency is the most common enzyme-related glycolytic effect. So pyruvate kinase catalyzes reaction number 10, where phosphoenol pyruvate is converted into pyruvate. This disease affects the R-type of pyruvate kinase isoenzyme, and this is the type that you find predominantly in red blood cells. So without this enzyme that catalyzes reaction 10, not only is that going to affect ATP generation, but it's going to lead to the accumulation of phosphoenol pyruvate. But if you see this reaction before that, it's reversible, so that can lead to the accumulation of 2-phosphoglycerate. And again, you see the reaction before that is reversible, that can lead to accumulation of this, and so on. It can lead to an accumulation of this and the other intermediates. So this disease is not only associated with decreased ATP levels in the red blood cells, but accumulation of many glycolytic intermediates. So patients with these red blood cells, they show very abnormal red blood cells, and these abnormal red blood cells cannot make it through the spleen. So many of them get destroyed in the spleen, and that leads to anemia, very low red blood cell count. And since red blood cells carry oxygen, and without enough red blood cells, of course oxygen is very important for energy generation, the patient is going to show fatigue, shortness of breath or dyspnea, and of course without enough red blood cells, they will have a pale skin. And once again, the breaking of red blood cells is going to release, the hemoglobin is going to be converted into bilirubin. So this increased bilirubin, which is yellowish in color, is going to lead to jaundice, that is the yellowing of eyes as well as skin. So these examples clearly show how important these enzymes that catalyze glycolysis are for that particular cell. And if that enzyme is absent or defective, then that could lead to all kinds of problems with that cell and eventually all kinds of problems in the body. This will also be a good time to talk about this deficiency called bisphosphoglycerate mutase deficiency. Now, this enzyme bisphosphoglycerate mutase is not a glycolytic enzyme, but it is very closely related to glycolysis because it converts one of the intermediates, 1,3-bisphosphoglycerate, into 2,3-bisphosphoglycerate, 2,3-BPG. Now in video 3.9b, we talked about the importance of 2,3-BPG. 2,3-BPG basically binds to the central cavity of hemoglobin. It stabilizes the deoxyhemoglobin form, the HHG form, shifts the curve to the right, the hemoglobin curve to the right, and that means the hemoglobin releases oxygen much easier when the curve is shifted to the right. So basically 2,3-BPG allows the hemoglobin to release oxygen much easier at the tissues. So clearly if this enzyme is defective, we are not going to get enough 2,3-BPG, and with a decreased concentration of 2,3-BPG, we are going to affect the release of oxygen from the hemoglobin at the tissues. So basically the curve will go more to the left. Now, if the tissues are not getting enough oxygen because of this deficiency in 2,3-BPG, then the body is going to respond by making more RBCs. That's called erythrocytosis. So now the person will have, this afflicted person will have a high RBC count. And that excessive amount of RBCs in the blood is going to lead to a very sluggish blood flow. And this sluggish blood flow can lead to symptoms such as headaches, high blood pressure, and dizziness. Now it's interesting to note that at this point here, if any enzyme that's catalyzing reaction 7, 8, 9, or 10, for example, we talk about pyruvate kinase deficiency, if there's any of these enzymes, 7, 8, 9, 10, that are deficient, then this will back up and we'll have accumulation. We'll have accumulation of 1,3-BPG, and that accumulation of 1,3-BPG will lead to higher amounts of 2,3-BPG. And these higher amounts of 2,3-BPG is going to shift the curve more to the right, and that will increase the oxygen release to tissues, just the opposite of what we see here. So hopefully these examples will show how biochemistry, including glycolysis and many other things we'll talk about in the future, is very closely related to physiology, and then which is eventually related to pathophysiology. So with this, we have completed the first part of our main street. In the next video, we shall focus on the second part, the pyruvate dehydrogenase complex, PDH. So until the next video, goodbye and have a wonderful day.